Welcome to the Masterclass on Fusarium wilt of banana. For this session on the history and epidemiology of Fusarium wilt, we have with us Wayne ONeill and Kenneth Pegg of the Queensland Department of Agriculture and Fisheries. And so I'll start by introducing Kenneth Pegg. He joined the Department of Agriculture and Fisheries in 1956 and has had a distinguished career as a plant pathologist working in horticultural research for 45 years. Among his many highlights are the development of a successful strategy to manage bacterial wilt of ginger and a groundbreaking work to manage Phytophthora disease on avocado and pineapple. In the banana community, is mostly known for his research on Fusarium wilt of banana. He's led international project on the diversity and distribution of the pathogen, assembled a large collection of isolate that has proved valuable for the development of diagnostic tools. He's led work on the evaluation of cultivars in race 1 and subtropical race 4 infected sites, received several honors as a fellow of the Australasian Plant Pathology Society. Even though he retired in 2001, he's continued working as an emeritus scientist, helping mentor young scientists and turning his mind to new plant disease problem. He's currently spending most of his time on studying the death of Araucaria in the Australian rainforests. When TR4 was detected in Queensland, his depth of knowledge and practical experience proved invaluable in formulating the response to the incursion. Thank you, Ken, for joining us. And then our other experts is Wayne ONeill. He started working on Fusarium wilt in the mid-1990s, performing diagnostic for the cotton industry using the VCG technique, a skill he later transferred to Fusarium wilt of banana. He's been part of the soil health and nematology team since 2005. He diagnosed the first detection of TR4 in northern Queensland and published the first record of the incursion. He was seconded to Biosecurity Queensland for 2 years to perform the VCG diagnostics and to investigate alternative hosts of the fungus and the effectiveness of containment strategies. He's currently working at looking at the possibility of using plant volatiles to detect asymptomatic plants. Thank you, Wayne, for being with us, for joining us. Okay, so I'll start with the first question to you, Ken. Because you work on several plant disease and you've developed management strategies for disease like Phytophthora disease, whose name translates to plant destroyer, so it sounds very ominous. So before we go into the specific of Fusarium wilt of banana, I'd like to have your view. How does it compare to other plant crop diseases when it comes to managing susceptible cultivars? Is it as inherently intractable, or is it the context in which it's grown in export trade production system? It's just for when you read extension stories on TR4, it seems very bad. But is it? You see,
when I was working on bananas, the industry where we had a Fusarium problem was in our ladyfinger variety, which is an A-B, and race 1 was devastating in that particular industry. But it was only a Queensland or Australian-based industry without any export. But it was a very, very serious disease. And then I worked on subtropical race 4, which turned up in Cavendish in the subtropics. And again, it was difficult to stop, but and eventually it took off most of the plantations. But now we've got a much more aggressive organism to deal with, and that's our TR4, which first appeared in Australia in 1998 in the Northern Territory. And there's only one farm left now in the Northern Territory. But in North Queensland, using the system we were talking about for holding the COVID-19, we've been able to hold TR4 very, very well. And I think Wayne has told me that so far we've only had 93 diseased plants. So it can be slow. You would never be able to eradicate it, but it can be slowed down. The spread can be slowed with good strategies of identification, containment, good surveillance, and then good quarantine measures. And a lot of the other farms now have biosecurity. And with that whole system, the pathogen has not got out of control. It's still under biosecurity control. But it's probably no worse than, say, Phytophthora was in avocados when I worked in that. That was a devastating organism. And it's only once we just found a control for Phytophthora root rot that the industry expanded and became a major horticultural industry. And it's very similar with TR4.
Okay, thank you. Guy, do you want to ask a question? I just want to, the newcomers, you can, if you want to ask a question, turn on your camera or put your question in the chat room. Yes, Guy?
Yes, thanks Wayne and Ken for a very good presentation. I had a question about the application of urea to control the fungus, and my question was more, although it's highly effective in significantly reducing the amount of fungus, would it also work when you have large numbers of plants that are infected, if it's across hectares and hectares of land? Because maybe it may be easy to apply it if you only have very few plants or mats that you want to treat, or where you want to treat the soil. But would it work on larger areas of land?
Would you like me to answer that, Ken? Yeah, well, we certainly wouldn't recommend that approach. The strategy we've developed is just purely for new disease foci. Its advantages are that it's very cheap. It's a simple method. You're not dealing with toxic chemicals as you would with other fumigants. However, in Australia, for example, we have a lot of problems with excess nitrogen use and nitrogen runoff, and that affects our Great Barrier Reef. I'm sure there are similar environmental problems in other parts of the world. So purely on an environmental basis, we couldn't recommend it. It's a very high rate, and I don't know how successful it would be on a large scale in any case. The horse is probably bolted by that stage when you've got a large infestation. We're treating the diseased area in a 10-metre radius. So around one plant at present, they're treating a 10-metre radius, and we feel that is doing a very good job of knocking back the inoculum in that localised area. We're not seeing, as far as I'm aware, we're not seeing any, after these patches have been treated, we're not seeing any spread along those rows or in those areas. New patches that pop up are in new disease foci from previous movement of the pathogen. So it is effective on these hotspots, but in short, no, we wouldn't and probably couldn't recommend it as a large-scale treatment. Anything to add to that?
I think that's why it's so important for this rapid identification and then going hard on that infested area and just treat that. But it's not effective if the soil moisture level is above field capacity, because with that water in the soil, a lot of it is ionised to the ammonium ion, and that's nowhere near as effective as the ammonia gas. So you don't want too much water there. You don't want excess water in the soil. You just want it nice and moist without and below field capacity.
Mm-hmm. Yeah, thanks. Very clear.
Thank you.
Okay, thank you. Miguel has a question about the first, when we started hearing about TR4, not sure, Miguel, if you could explain that it originated in Taiwan in the 60s, but we learned it was in Indonesia in the 90s.
Yeah, I'll just read that out for Ken. He just, with since his operation, he won't be able to read that at the moment. But Miguel is saying that we first heard about TR4 on a widespread basis, I guess, from when the export plantations in Indonesia started becoming effective. However, now in retrospect, we think that what was causing disease in Taiwan as far back as the 1960s was also TR4.
And also in China in the early years.
Yes, yeah. So I think it was originally, when disease was occurring in Cavendish in Taiwan, it was originally written off as being something similar to the subtropical race fall, what we now call subtropical race fall, which is seen in the Canary Islands in Australia and South Africa and a few other places. However, when we got the technologies to identify the actual strains of the fungus, it turned out that the same BCG that causes TR4 in most places around the world predominates in Taiwan. There is also BCG 0121, which is another closely related strain, and as well as what we would call a subtropical race for strain in Taiwan. However, what predominates and what is causing most of the disease we now know is TR4, and that's probably what caused the start of the epidemic as far back as the 1960s in Taiwan. It's likely that these strains originated in, as I said in my presentation, in peninsular Malaysia and Sumatra. And so some of the early planting material that was taken to Taiwan perhaps came from that location. Perhaps that's how it first got to Taiwan. So in short, there are subtropical race fall strains in Taiwan, but the predominant strains that are causing disease are what we now know as TR4. I think it's just a matter of the technology wasn't available to discriminate those strains at the subspecies level back when that epidemic started.
And what do you think of the Budenhagen's theory about the coevolution of, well, TR4 or other strains with wild bananas and then being moved out? Is that what happened in Indonesia, in Malaysia?
That's what we believe, that it originated in those countries, co-evolved with the wild bananas, and it was disseminated by man through all the various areas. But Ivan could never find any native bananas that were infected with TR4, but it was there. That's how we think it developed. And it really took off when they started using tissue culture plants for planting. The nurseries did not use pasteurised soil, and they planted all these fields. There was no hygiene, and then the disease started to appear. And instead of acting quickly, they sort of just admired the new disease, and it spread rapidly, and now it's managed to get to almost every country in the world. I think it probably came into northern Australia probably a long ago, because the Macassan people used to come down from Southeast Asia into northern Australia and do farming. Now, the first outbreak we found in the Northern Territory was close to an old Macassan well, and that was then planted with bananas, and they were using bananas as a shade crop for mangosteen, and so that's how it got into Australia, I think. And then it's eventually found its way into North Queensland, and then it's spreading around the world. There's hardly a country without it now.
But yeah,
it did co-evolve with the wild banana in the equatorial areas of Malaysia and Indonesia. Whereas I think the race ones probably originated more in the India, Borneo, Vietnam area. They're the race 1 VCGs. But I think the race 4 VCGs definitely co-evolved in Malaysia, in equatorial Malaysia and Indonesia.
You wanted to add something, Wayne?
No, that's Ken's pretty much summed it up. But yeah, I would agree with Ivan that yeah, they probably co-evolved. Local wild bananas were probably, you know, very tolerant of the local FOC strains or whatever they were at that primordial stage. But it was only when people started selecting, you know, parthenocarpic bananas for edible bananas and started moving them around at first locally, and then from island to island, they would have then been exposed to strains that they weren't naturally exposed to, and yeah, and then disease started, I guess.
But that also that means that in a resistant banana, what is pathogenic to a cultivar and lives on as an endophyte in a resistant banana, like the Cavendish in Latin America would have it's resistant to the race 1, but those race 1 live as endophyte in Cavendish. And then when you're an endophyte, what I'm asking, because somebody is also asking a question about the chlamydospore. When you are an endophyte, according to Buddha-Nagan's theory, and you are in the banana, this is also the issue of alternative hosts. You undergo your life cycle as if you were a pathogen. It's the same, and then you produce chlamydospores and spores. Is that how it survives in the soil?
I know that, well, some workers at the University of Queensland, they used some GFP transformed isolates looking at alternative hosts, and they did visualise chlamydospore production on those alternative hosts. However, the colonisation seemed to be quite reasonably superficial, but they did see chlamydospores. That's correct, isn't it, Ken? So yeah, it is completing its life cycle, if you want to put it that way, in producing those resting structures on the alternative hosts, and perhaps the same happens for resistant or tolerant bananas with strains that don't cause disease.
With endophyte so I don't know if it answers the question. And their research on chlamydospore as they are the reservoir of the pathogen inhabiting soils to banana culture. So is there any yeah, I guess it's to maybe try to I don't know what kind of research particularly, but these are the ones that are problematic in the soil. Is there any way that they seem to survive? What I'm surprised on is that a fallow doesn't seem to get rid of them. They seem to be decreasing. So how do they survive if they're not in a host? So if you remove the host and they still survive, does it say it's because they're saprophytes?
No, they're a resting spore. They survive. A lot of fungi have chlamydospores that are really, they don't survive as long as oospores. There's no oospores in Fusarium, but other organisms have oospores. So they're a survival organ of the fungus, and it's probably short term. But the fungus also survives in the alternative weed hosts, weed and grasses. So that's how it survives in the soil. Chlamydospores will be there, and they'll survive for a long time in decaying banana tissue or be released into the soil. But they're probably not going to survive for years and years. So for long term, I think you need these alternative weed hosts or herbaceous weeds or grasses.
Yeah, that's the thing. In the absence of alternative hosts or banana hosts, nobody knows how long they would survive in the soil, since fallow seem to reduce the the concentration of chlamydospore, but they don't
Yes.
die eventually? In the lab, apparently they die fairly quickly.
Yes, yeah, I don't think we have good data on that from the field, and that is a gap in research. I mean, traditionally they were ascribed those really long survival times of 20 or 40 years. From the work we've done more recently, it appears that alternative hosts probably play a bigger role in the survival, and some of those fallow situations that were reported on may not have been truly bare fallows. I think if you had a truly bare fallow, you are going to reduce the inoculum within a matter of years. I believe in the Northern Territory, they had a trial site there. They had a reasonably bare fallow situation, and also combined with they've got a relatively dry climate there and very high temperatures, they found that their inoculum was fairly dramatically reduced within just a couple of years with a fallow situation.
Get back to that. There's a question: can alternative hosts such as the asthma weeds host FOC without bananas around in a particular area? How long, despite no bananas around?
Yes, asthma weed was actually one of the ones we tested in our work. I'm just looking it up in our guide now. Yeah, TR4 was isolated from asthma weed in the Northern Territory from a field situation, and also in our glasshouse inoculation studies, it got a medium rating there. So it was also able to host. That was subtropical rice borer, I believe. No, sorry, that was also tropical rice borer in the glasshouse. So it was in an inoculation study. It also hosted the pathogen. Where was I going with that? It seems to be related to inoculum levels. Like in inoculation studies, it seems almost anything will host the pathogen if the inoculum levels are high enough. In field situations, we tend to find we really have to look. You know, it seems the more we look, the more we find in a field situation. But we were sampling close to symptomatic plants, so obviously areas with high inoculum. So in that situation, it seems that almost anything has the potential to host the pathogen. But in our inoculation studies, there is a gradation. So some things seem to be better hosts than others, but almost anything has the ability to host a pathogen when inoculum loads are high. And I think the pathogen will then just tick over. It won't lead to any great buildup of inoculum in the field, but it will allow the pathogen to survive in the absence of a banana host.
This is just a thought experiment, but well, that's sort of the factors that I was sort of alluding to at the beginning when I say how intractable it is. I mean, that the fact that it can be any host can keep it going. Could it? But it's the exudate of the host produced by the host and the roots of the host that stimulate it to germinate, and that's when it enters the plant.
Yes, yes. And so possibly also these alternative hosts would have perhaps some of the same exudates and would also be stimulating the spores to germinate.
But if you could get the spores to germinate in the absence of the host, but I guess it's not practical. But then would they die quickly? But that
I guess like as a trap crop sort of situation, is that what you're thinking?
Well, there doesn't seem to be any crop that would not be a host.
Yeah, yeah.
Well, although
I mean,
some seem to have antifungal abilities.
But yeah, coming up with the right chemical to make those spores germinate and then having a bare fallow, something like that. Yeah. But yeah, just getting back to the intractability, and anecdotally, where I live, which I showed an old photograph of the valley where I live, there were bananas growing there in the 1920s. They were gone by the 1930s. I planted tissue culture bananas there, well, 10 years ago now, but they became infested with Panama disease almost straight away. The fungus was still there in the soil. What's that? 90 plus years later, with no bananas in the intervening years. So just that would have been survival mainly on alternative hosts, I guess.
Yeah, and in Latin America also, hear the same stories about, and in the export banana production system, which are, besides banana, they don't seem, I don't know, maybe that has changed, but they don't seem to have a lot of other alternative hosts because they're usually weeded. And they still, after 80 years, if you try to plant a Gros Michel, the race 1 strains are still in the soil after 80 years.
Mm.
And but it also speaks to the resistance of the Cavendish, which I find amazing, because
Yes,
I think it hasn't broken down the resistance. Is it because the fungus reproduces asexually? Is that usually what happens with those types of fungus, that they don't break down the resistance of their host so easily?
You've got a clonal host and a clonal pathogen,
Yeah,
largely.
it's the speed of the reaction, isn't it? That the resistant plant will block it very, very quickly and not allow it to become systemic in the plant. It can be there in the roots, because it will stop the further development. Within the Cavendish against a race 1, it may enter the roots, but it won't get much, it probably won't even reach the core.
Yeah, yes, that's the mechanism, but the pathogen doesn't seem to be able to overcome and to mutate to overcome the resistance of the Cavendish. Yes, we have a question for Miguel. It seems that weeds and grass play a more important role on FOC survival than previously recognized, but bare soil can also boost, epidemics. Tony Patterson's presentation explores it, and we will learn more about that on Thursday. Okay, so it's not so much a question, but
goes into more detail about the practical issues of this in his presentations.
Yeah, okay.
The balancing act between the benefits of having vegetation in your interrow area and the fact that they also act as alternative hosts, and the benefits of having the right species there, I guess, will outweigh any disadvantage of having alternative hosts there.
Mm-hmm. Yeah, it's really a balancing act. Huh?
To answer your previous question about race 1 never attacks the Cavendish, it will occasionally if the plants are stressed in some way. Wayne had an experiment that was where the roots were heavily infected with burrowing nematode, and he had a plant there succumb to race 1, and that was a Cavendish. Also, Cavendish will succumb to race 1 if your soils are waterlogged or growing in an acid soil. They can succumb, but the host has to be very stressed for them to invade. So it can happen if the plant is under extreme stress.
Yes, and that's what happens also, the STR4, the subtropical race 1 you have in Southeast England. But you seem to be saying earlier, Ken, that it did cause a lot of damage. Is that because usually it should be easier to manage when it's a subtropical race 4?
It's not easy to manage in the subtropics. It does not cause disease, but it does cause disease in the subtropics, where Cavendish suffer from the cold weather. So they're under cold stress during the winters, which they don't get in the tropical areas. And so it spreads in exactly the same way. And particularly if you have poor soils, cool weather, it can cause quite a bit of considerable damage.
I read, Jeff Daniels sent me an article after he saw my presentation, and he said that in the Canaries, they've actually been able to manage it quite well there. They had big problems back in the 1920s, I think it was, but that was mainly related to acid soils. Their temperatures there are sort of more moderate and, more moderated by being an island, I guess. So they don't have as much cold stress in the Canaries compared to here. They've got the same VCG of the pathogen, but once they were able to sort of ameliorate their soils to some extent, the disease was largely controllable. It's not it's not nearly as significant there as it is here, even though it's the same strain in the subtropics.
But I think it's, yes, Ken?
Our bananas in the subtropics, there's a lot of chlorophyll degradation in the winter. The plants actually turn yellow, so they are under enormous stress, and so they're really cold stressed, and under those conditions, the STR4 becomes quite a serious pathogen.
But so they're still growing bananas in southeast Queensland under the where there is the STR4, so they still manage it.
There's very little, but there's a whole range of issues. The price of real estate has been one of the main factors for people not growing bananas here, because the land is too valuable and it's been sold for rural lifestyle type living. So there aren't many farms left in southeast Queensland. There's still quite a large amount of bananas grown in New South Wales, which is just over the border. That's south from here. But they don't have as much subtropical race 4 there. It's not as widespread. It's mainly race 1 they're dealing with there. But still, even with race 1, it gets so bad that growers they're forced to switch to Cavendish.
But that's it also, those strains don't seem to move as easily as TR4, because I mean, I think it's during the presentations, you say that you'd be interested in seeing how a subtropical race 4 behaves in tropical condition, but it might be there. How do you know? You don't have a molecular test to detect it. So why didn't it move to Queensland?
Sorry?
Why didn't it move to the North Queensland, where the conditions are more tropical?
Well, we have traditionally had fairly good quarantine zones even within Queensland. The industry in North Queensland has been fairly well protected, and well, purely by geographical isolation. It's thousands of kilometres away, but we've also had restrictions in place by legislation to prevent movement.
Yeah, no planting material was allowed to move to North Queensland from the south. So control of planting material, it was never moved to North Queensland properly.
There's a question waiting, but I just want to finish this. But do we know that the TR4 in North Queensland, is it the same as the one in the Northern Territory? Can you do that sequencing to see if they're the same, or was it a reintroduction from outside?
The origin is not known, whether it's a fresh introduction from overseas or whether it has come from the Northern Territory. Certainly they're the same BCG. I know that with sequencing really recently, they've been able to find very SNP level differences between TR4 in different parts of the world, but I don't think there's been enough evidence to link that introduction to the Northern Territory or as a fresh introduction from overseas. So it's unknown at this stage.
Mm.
There's so very little variation within the pathogen. It's very hard to do that kind of tracing, even with sequencing data.
Yeah. Guy is asking if there are any cultivated crops that could be used in rotation to reduce the amount of inoculum in the soil.
Yeah, I'd say hold this space and wait for Tony's presentation later in the week. But as well as looking at alternative weed hosts, we have done experiments looking for rotations that might be useful. And the short answer is that we've got an announcement here. Just bear with us for a second.
If you wish to work back to row 200, please come down Sunday after to register, which is like a secret test underground for. Thank you and have a good evening. Okay, sorry about that.
No
yeah, we also looked at potential rotation crops, and as with the weed hosts, there is a gradation in their ability to host the pathogen, but we certainly haven't come up with anything that is a good non-host rotation crop. That would be ideal if we could identify something like that.
In the Northern Territory, Wayne, don't they alternate with pineapples?
Yes. Yeah, the one commercial farm that Ken mentioned earlier that's still going in the Northern Territory, they grow Cavendish for 2 cycles, I think it is, and then they'll rotate to pineapple for a couple of years. So we haven't tested pineapple as a rotation crop in its ability to host the pathogen, but that system seems to work reasonably well. Its inoculum obviously does reduce to some extent under that system.
But what happened in Northern Territory when it first arrived? Why did it got out of control? Because Australia always had a good reputation for biosecurity. That was in 1997 and was first detected in Northern Territory.
Well, I actually visited that farm in 1997. And when I went there were a lot of holes in the plantation where the plants were missing and the soil had been disturbed. And I said to the grower, "What's going on here? What have you done?" And he said, "Well, as soon as I see symptoms, I dig out the plant, put it on my tractor and dump it in the bush." So there was no biosecurity involved whatsoever, no hygiene. So it was disturbing the soil and dumping the plants in the bush for the first outbreak. That was the one near the Almacassin well that we referred to later.
Mm-hmm.
So that's the way he had treated his original outbreak. You know, it's not his fault. He didn't know at that stage that he was doing the wrong thing. But there was that, first of all, disturbing the soil, and that's what we never do in the TR4 when we have a plant with TR4. We don't disturb the soil whatsoever. We just throw we don't dig in the urea. The urea goes on the surface of the soil. It's not dug in. So he disturbed the soil, was moving his tractor around through the plants and then dumping plants in the bush. So that could have been the start of that epidemic in the Northern Territory.
Yeah, it just probably wasn't caught as early as North Queensland. And Ken and I were talking about this earlier. There's a bird in the Northern Territory called the magpie goose, and they can appear in large numbers, and they'll go foraging through crops and things like that. And it's even possible that birds like that could have, you know, got the pathogen on their feet and then moved it from place to place as well. And like North Queensland, there's also wild pigs, so there are sort of vectors that are beyond our control. There are animals and birds and things like that that can potentially be you know, spreading the disease around uncontrolled. So people have speculated about things like that for the, you know, how it was rapidly moved around in that case.
Yes. And somebody is asking a question about suppressive soils. I mean, there are suppressive soils during the epidemic in Latin America of the race 1 on Gros Michel. There were known suppressive soils. Is there any recorded for TR4? Suppressive soils that are suppressive to TR4?
Not to my knowledge. Ken, suppressive soils to TR4? No.
We certainly have suppressive soils for race 1.
Yeah.
It's the slow wilt soils, but I don't think there's any for TR4.
Yeah, and perhaps that comes back to...
It's probably too aggressive.
Yeah.
The organism is too aggressive.
Too aggressive, yeah. What does it mean exactly? Is it too aggressive in relation to the host, or because suppressive soils supposedly other microorganisms that keep them in check? I don't know. Nobody exactly figure out how they work, and it's certainly difficult to replicate. Nobody has replicated, so.
Well, again, Miguel's saying that a lot of this will be discussed on the 3rd day, so we won't go into too much detail. But yeah, Tony's been working with a group at the University of Queensland who are looking at the soil microbiome and what attributes are associated with microbial suppression. So I guess there's a lot more information to come on that. But yeah, to answer the question, as far as we're aware, there's no recorded suppressive soils for TR4 at this stage.
Mm-hmm. But this question of aggressivity, just how is it defined? How is it measured? What's so special about TR4? If we knew what was different, would it help in terms of control measure? I don't know. It's a bit.
Well, it gets down to the lack of host resistance, I guess. But yeah, in terms of the actual mechanisms, that's not my field.
Yeah. Do you think there are other strains out there that could pop up, or do you think we're pretty good overview of the existing? There doesn't seem to be, again, this question of horizontal transfer. They are the strains, I guess they don't have all the same origin. They are the strains that co-evolve with the host, but there seems to be also the, you hear about horizontal transfer from Fusarium fungus that attack other crops. Is that an important mechanism for creating pathogenic strains to?
But there are probably still a lot of strains in Indonesia and Malaysia that have not yet been found.
Yeah, and from work going back to the 90s that Ken did with Susie Perry, Susie Bentley then, they ended up with a whole lot of undescribed genotypes that didn't match with existing BCGs, and there has been more recent work where just in publication at the moment where BCGs are being ascribed to some of these genotypes that were never allocated a BCG, and I believe that other isolates have been collected in other parts of the world now that are also being given new BCG
new strains.
Yeah, so yeah, there are more and more, at least at the BCG level, there are more strains out there.
So in the year 2000, there were 20 BCGs, and I think now there's 40.
Well, I think that went up to 24, and there will be a 40-ish shortly.
Mm-hmm.
So there is a lot more diversity out there in the pathogen.
And a lot of these are from strains that have been collected in the past. And because they didn't fit into an old BCG, just put to one side.
And I guess that raises the question, if some good resistance is found to TR4, what will the pathogenicity of these other strains be? I mean, we ideally want a banana that's resistant to all strains. So if resistance is found to TR4, it will have to be screened against all known strains to be, lest we end up in another epidemic situation.
Although TR4 seems to be the strain that is, although I'm not sure how it compares to race 1 on Gros Michel, you seem to be saying that TR4 on Cavendish is more aggressive than race 1 on Gros Michel?
Oh, no, I think, well, Ken actually said earlier that race 1 in the ladyfinger variety here can be as bad as what we see in TR4 with Cavendish.
It can be just as severe.
Yeah.
On our AABs, that's race 1 BCGs, can be as severe as TR4 is in Cavendish. But TR4 is much more aggressive than subtropical race 4, STR4, in Cavendish.
Yeah, I guess you don't have Gros Michel in Australia.
No,
to test that. But when you look at the epidemic in Latin America, the race 1 on Gros Michel, that was very severe. What were they? Obviously, what they were doing didn't help either, but it seems comparable.
Ladyfingers was also very severe. And the way they slowed that down, as soon as they detected an infected plant in their plantation, they would immediately cease irrigation. No more irrigation on that block, because that stopped the fungus contaminating the water and being recirculated around the farm. So they dare not irrigate once they found that Panama disease. And that's how they slowed it down, slowed the epidemic. But and it was never a mechanised industry. It was all handwork, and so they could control movement. And there was no movement of soil from the infected area, which you cannot do in the big plantations these days.
Yeah, here in the subtropics, we grow bananas on slopes to avoid frost damage. So it's not a very mechanised industry, as Ken just said. Whereas in northern Australia, it's grown on flat land and it's much more mechanised. So there's higher potential to spread the pathogen around more quickly that way.
But in Latin America, they were spreading it because they were opening new lands also. That would have been the main driving force of the epidemic at the time, because they were replanting with infected material.
Yes, yeah.
Not so much because they spread it. So what do you see the pattern in you have very few farms that are infected so far, but do you see a pattern of how the fungus moves around between the 5 farms that is it 5 farms that have been infected in Queensland?
Yes, well, unfortunately, a lot of the movement occurred before symptoms appeared on the first farm. So a lot of what we're looking at could be historical movement, because there are contractors that go from farm to farm for various reasons, and some of the spread may have happened that way. But another problem this is in the Tully Valley, and it's the highest rainfall area in Australia. And the Tully River, which flows down the centre of that valley, it partially inundates some of these farms. So there's obviously going to be some pathogen spread with floodwaters. As I mentioned earlier, there's also feral animal problems. There's wild pigs that move from the rainforest onto the farms and move from farm to farm, which is another uncontrolled means of spread. So the Biosecurity Queensland are very big on contact tracing. So they did extensive tracing, trace back and trace forward when the first incursion occurred, to rate the farms in terms of their risk of whether the disease will appear on their farm, and they've used that to inform their frequency of surveillance on properties. Farms that were considered higher risk because they had more connections received more surveillance. So I guess that's helped, but yeah, unfortunately, a lot of the spread, or some of the spread, may have occurred before symptoms appeared on the first farm. And also there's those uncontrolled factors like flooding and wild animals and things which we can't really do that much about. So yeah. So when it appears on, sorry, the disease foci on these farms are often quite random. So it doesn't seem that we're seeing spread from the patches that we've already picked up. It seems that it's some spread that's occurred in the past through movements of machinery or animals or something like that.
So the first farm where they saw the disease was not necessarily the first farm to be infected. So it evolves that the symptoms appear at different rates depending on the condition.
We think that the infection had been on the first farm that was detected for a couple of years before it was realised what was going on there. In the intervening years, while the pathogen is there in the soil, these contractors are going from farm to farm, and yeah, so it's spread that's happened in the intervening years between the pathogen arriving and the first symptoms appearing, is what we would surmise. Do you agree with that, Ken? Yeah. And some spread has certainly happened since then with, as I said, with floods and things like that, which are uncontrolled.
But I read somewhere that it seems to be moving upstream. Is it the case? Not the moving, but the infection site seems to be more and more upstream.
I think it's going. I've been out of biosecurity for Queensland for a little while, but certainly the second and third properties that were detected were upstream of the first property. Yeah, so they probably represent some of that past movement.
Movement, but by land, not by
flood. Yeah.
By flood, yeah. We have a question on the classification of the strains and races, but it needs to be revised. Now we've heard of a new name species that have been proposed for TR4 and race 1. Do you have any thought of that on that?
Well, I do not accept the new name for race 4. Was they called Odoratissima? It wasn't Fusarium. So what they've done, they've elevated from the former specialis to species, and they've used that name, Odoratissima, but for TR4. But subtropical race 4 isolates also produce that same volatile. So where does that leave them? How do you distinguish? These subtropical race 4 also produce the volatile as TR4. So it's a very poor choice of name, and it adds to the confusion. And also they haven't related these species with the they haven't related to anything, have they? Not sure.
Oh, it's based on some morphology and some host reaction, I think. But I think certainly for a long time we've known that FOC is comprised of likely cryptic species. So yeah, I think some form of revision is probably needed. There's certainly always been, no matter what sort of analysis you use to look at the pathogen, they've always fallen into those two broad groups of the race 1 type isolates and the race 4 type isolates. And they certainly, the isolates, the strains we have in Australia, they actually look different morphologically. We can tell what they are just looking at a plate. So there's probably some need for revision there. But I think greater discussion and consensus is needed. Otherwise, we're just going to end up with more confusion. The race system itself is very confusing. I think that's fairly unworkable. But all of the sort of VCG by host variety, by environment reactions are all quite unique. So it's such a diverse pathogen and such a diverse host that it's really hard to know where to draw those boundaries usefully.
Now, instead of having CGs, you ended up with 9 species. And it doesn't mention Bioversity any longer.
Well, that's just the one they identified so far. There's more to come, no doubt. Do you have some insight on the spread of TR4 in China and the Mekong Delta? Don't know if you've been following the situation.
Personally, I haven't been involved with that, but from what I understand, some of the large companies from China have been expanding their operation into some of the countries like Laos that share a border. So yeah, so I think it's just spread with farming operations across those borders. But yeah, I personally don't know much about that area.
Ken, when you see the research, you were there in the you did a paper recently going back over all the research that was been done on the epidemiology of the disease, mainly by the United Fruit Company and also by the British scientists. What do you think are the questions now that should be answered, that scientists should look into it, but they are not? What do you think of the current research now? What's going on? Are they asking the right questions, or what do you feel are priorities that should be looked at that we don't know, or are they reinventing the wheel? Are we redoing a lot? Are the scientists doing a lot of the same studies that were done in the past? What's your take on that?
There's a lot more new technology now that you can use. You know, all the molecular technology that we didn't have in the early days, and the effector genes, all that work is now known. But now the concentration has to be on finding a resistant plant. Resistance is the best solution to the problem. And perhaps it'll have to be GM, or it'll come from CRISPR technology, the new molecular technologies. But I think the whole world, what they have to do is have a new resistant banana. Otherwise, the control will never be complete until they do that. And there are already GM bananas that are resistant, and they're Cavendish, that have been genetically modified. Now, of course, a lot of countries won't take genetically modified bananas. And also the gene editing, the CRISPR technology shows a lot of hope. And I think the Malakensis gene, which has resistance to TR4, is already in Cavendish, but it's dormant and it just has to be elevated up, switched on. And I think that's where the whole solution will eventually come from.
For the export trade, but if you want to grow a susceptible cultivar, it gets complicated.
Yes.
Yes.
Then you've got to get all the work that Tony Patterson's doing.
Yes.
And use a microbial system.
Yeah, so integrated management is the other way to go in that sort of situation.
Yeah,
But that will never give complete control. If you want complete control, you've got to change the variety.
Mm-hmm.
And I think Tony's going to probably talk about this, but when I was working when we did our work in the 1980s, 1990s, we worked very closely with the plant breeders, and we used to screen their diploids and also their tetraploids from Thea, CRAD, Improper in Brazil, and we had their material we used to screen. And out of that, the tetraploid, I found Goldfinger was highly resistant to subtropical race 4 and race 1, and has also been found to be resistant to tropical race 4. And I think Tony's team are mutating it to get better fruit quality. But that did show high resistance. And that was one of Phil Rowe's breeding, which he bred for the Third World, and a very vigorous plant, resistant to Sigatoka's nematode, a magnificent plant. But the fruit quality wasn't what the market wants. But it does have resistance to TR4.
Yeah, that's an interesting strategy, which again, maybe Australia, because it's a domestic market, can introduce a new variety for the export trade. They still pretty much want something similar to Cavendish. That's another hurdle. In your presentation, you talk about the risk of the fungus moving with because it's in the peduncle and the cushion also. So do you think that's a high risk for the export trade? Like I'm thinking of Latin America, if they start if they have if a TR4 spread, but so it could be with the bananas that are exported, but if it's in the peduncle, but wouldn't you see it before?
Well, it's very unusual for our plant that has Fusarium in the peduncle to produce a marketable bunch. Normally the bunch is very inferior. But Wayne knows a bit about it being in the cushion. I'll let you talk about it.
Well, I know it's certainly been isolated from the cushion or the crown, and some of that material makes it into a carton with a cluster of bananas. So there is potential for it to be moved around with fruit. Here in Queensland, the farms that are still operating in the presence of the disease, they have protocols in place to minimise that risk, and you can certainly minimise it by not taking fruit from obviously diseased plants. Post-harvest fungicides can also help with killing the fungus that may be in that fruit. It's all that material associated with the fruit. With more and more tolerant varieties being grown in some countries, there's a greater risk that a higher proportion of infected plants will be producing marketable fruit, which you mightn't see in traditional Cavendish cultivars. So I guess there's a greater risk of the fungus, making its way up to the bunches if people are growing those tolerant varieties. So I guess it's something to be more aware of. But Yeah, I think there's some risk, but those risks can be minimised to a large degree of moving the fungus with fruit.
And you mentioned also that those tolerant varieties, if they are to be deployed, shouldn't be deployed too early because then they would mask the spread of TR4.
Yeah, well, that's one line of thinking. Yeah, I mean that's certainly a possibility.
Yeah, the early stages of the epidemic, while you're still in the lag phase, it's probably best not to use them. Once you get to the exponential phase, it probably doesn't matter.
Mm-hmm.
But in the early phase, perhaps you shouldn't do that. When you're trying to, you know, you've got to keep that population low, the inoculant population very, very low. Because once it escapes out of that lag, I don't think you'll ever stop TR4 once it gets into the exponential phase.
Yeah, I guess if you are trying to manage it in the early phase, seeing those diseased plants is the best indicator of where the pathogen is.
You don't want to mask its presence by using a more tolerant variety.
But should there be more testing on non-Cavendish cultivars that are susceptible to race 1? So when they develop Fusarium wilt, people don't know, assume that it's a race 1 when it could be TR4.
Yeah, I mentioned that as an issue of possible complacency. That, you know, if there are race 1 susceptible varieties, people always just assume, oh well, it's just another case of race 1. But we should be more vigilant with our testing, I think, especially if you're near commercial Cavendish production areas. There's always a possibility that TR4 will first appear in another cultivar besides Cavendish, and if it's ignored, then it can build up inoculum, you know, before it's realised what is actually going on.
And that's where we got caught out originally with subtropical race 4. We only found it when it was affecting Cavendish, but unknown to us, it was present in all the ladyfinger plantations with race 1. And we had assumed that all our Panama disease in the race 1 susceptible cultivars was due to race 1. But we later found it was subtropical race 4 was there causing disease with race 1. So it's important, if you're looking at any cultivar, to find out exactly what isolates are in there if you're having disease. So we had STR4 with race 1, race 1 widespread in the ladyfinger plantations, and we didn't know. We had no idea.
Yeah. What about your work to detect asymptomatic plants through their plant volatiles? Are the volatiles that are produced by the banana plant?
Well, it's just a scoping project at this stage. I think there's an Israeli group maybe that was doing similar work a while ago. I don't know how far they've got, but we've long known that the race 4 isolates produce these characteristic volatiles on a starch substrate. We use rice as a volatile test. It's one of our diagnostic tests. However, we didn't know if those same volatiles were produced in the banana plant. So years ago, Ken had a paper with Natalie Moore where they looked at the Australian isolates and which ones produced these volatiles, and they produced chromatograms and worked out what these compounds were. We've got better technology these days for analysing the gases. So we've done a glasshouse experiment where we inoculated plants with race 1 and race 4. We enclosed the plants in cellophane and then collected the gases and analysed them. And our chemist was able to pick up differences between inoculated plants and uninoculated plants in the case of race 4 before they had external symptoms. However, it wasn't that same volatile compound that the Fusarium produces on the starch substrates. They were plant volatiles, limonene and pinene and that sort of thing. I can't remember exactly all the compounds, but they were plant volatiles. But he was able to pick up a difference. So at the moment, we're taking this work to the Northern Territory just to see if we can repeat the results with TR4 because we can't work on TR4 in Queensland. How this actually translates to a diagnostic test, I don't know. I know there are some workers in the US who they've developed a little pump and colorimeter that you can even just attach to a smartphone, and what disease were they working on, Ken? I can't remember. I can't remember what the disease was, but they've successfully got this system where you can just take a leaf sample, pop it in a container for a short period of time, then you can use your little smartphone pump to extract the volatiles, and then you have just sensitized paper strips according to what your target volatiles are, and it integrates the phone camera as a colorimeter to look for a color change with your sensitized paper strip. So this technology does have potential as a field test, but how that would translate for Panama disease, I don't know. It's just a scoping project at this stage, but we have had some positive results.
Maybe dogs would be good. They seem to be good at detecting a lot of things.
Yes, indeed.
Somebody is asking about the incubation period from the infection of Fusarium wilt banana to the presence of symptom at the field level is important to establish management strategy. What's the reason for the long incubation period in banana plantation? These periods, infection in the presence of visible symptom, been well studied in the field?
So incubation periods and latency.
Yeah, the incubation period depends on how much inoculum is in the soil. That plays a big part for a start. When inoculum levels are low, you have a long incubation period. When they're high, you have a shorter incubation period. And they also relate to the plant itself and the plant stress. For example, you can have a beautiful looking Cavendish banana that looks perfectly healthy. It throws a bunch and very quickly shows symptoms of Panama disease. So it's the physiological state of the host as well as the inoculum levels in the soil that are involved in the length of the incubation period, as well as climatic factors and amount of rain and soil moisture. So I guess it's host physiology and inoculum pressure. So when your inoculum levels in the field are very low, you generally find only scattered plants that show symptoms. They take a long time to appear. But if you have a massive inoculum level, you start to get clusters of plants appearing, and the incubation period is much shorter. So as the inoculum level increases, the incubation period decreases.
I think we mentioned in our talk as well that talking about environmental conditions, the conditions that favour infection are not necessarily the same as those that favour disease expression. So you might have stresses due to drought or cold temperatures that favour infection, and then I guess that infection can sit as a latent infection. There might be, it might enter the roots, it might be sitting in the corm, but then you might not see that symptom expression until you get favourable growing conditions for the plant to grow rapidly, when there's lots of transpiration, lots of movement through the xylem, and then the fungus can quickly invade the rest of the plant. Or it might be the stress of bunching, as Ken mentioned, might be when you rapidly see symptoms develop.
Any parameters to check how much is farm affected by TR4? I guess, yeah, because you only see those that have visible symptom. There are tests to check. Is that about the inoculum test to check the inoculum in the soil or something? Is that?
Yeah, there are. I mean, some work has been done on molecular tests for soil, and some of them work reasonably well. But Ken's made the point that a positive result from a soil test is a positive test, but a negative test, you don't really know. Sampling is a big factor with, sampling. The number of samples you have to take, sampling errors is a big factor in how much use any sort of soil sampling could be. Provided you've got a good assay that overcomes any sort of inhibition from extracting from the soil and that sort of thing. sampling is certainly a big factor if we wanted to go to soil sampling and soil testing.
Okay, I don't know, is there any questions? Just want to give people a few minutes a chance to ask their questions. There's a lot of, I imagine farmers receive a lot of publicity by people who have a product to sell that will control.
When the disease first arrived in North Queensland, we were inundated with people who had the products that would cure the disease. So we actually had to, it got to the point where we had so many inquiries where we had to go to we went through a process with Ken of shortlisting these products based on whatever scientific merit they might be able to provide of, previous trial results, even in other crop or pathogen situations. So we did shortlist ones that showed, some sort of scientific merit. And then we actually conducted our own sort of independent glasshouse trials on the most promising products, and unfortunately, didn't come up with anything that suppressed the disease. No, some were, good growth promoters, but as far as stopping disease, they didn't work.
Yeah. Okay. Great. Maybe we can end here.